All about biochemistry. First off, atomic structure. To begin with, a proton is the first main part of an atom. Protons have a positive charge, and the number of protons in an atom identifies it as a certain element on the periodic table. Secondly, there are neutrons. Neutrons have a neutral charge. If the number of neutrons is the same as the number of protons, then it is an average atom. However, if the number of neutrons is greater or lower than the number of protons, then that atom is an isotope. Third, there are electrons. Electrons have a negative charge. If the number of valence electrons is the same as the number of protons, then it is an evenly charged atom. However, if the number of valence electrons is greater or lower than the number of protons, then that atom is an ion. Next up is bonding. Bonds are formed when two nuclei attract the same electrons. Bond forming then releases energy. There are several major types of bonds, and the first of which are known as ionic bonds. These occur via the transfer of electrons, as seen as this diagram. With the next type, covalent bonds, there are two subgroups, polar and nonpolar, but both are defined by the sharing of electrons between nonmetals. With polar bonds, also known as unbalanced bonds, water molecules are a great place to start. The oxygen atom shares the electrons of two hydrogen atoms, and as a result, oxygen becomes more negatively charged, and each hydrogen atom becomes more positively charged, creating a stronger attraction between the three atoms. With nonpolar molecules, also known as balanced molecules, the bond forms between two atoms of the same element and share their valence electrons. With metallic bonds, metallic atoms share electrons with one another by allowing their electrons to gather in one area between all the metallic atoms, creating a negatively charged area and a positively charged area. This in turn leads to greater conductivity and malleability. On to the properties of water. The properties of water are caused by the high polarity that water molecules have, as well as the hydrogen bonds that result from the attraction between the positively charged hydrogen atom and the negatively charged oxygen atom of adjacent water molecules. These hydrogen bonds, in turn, create a great deal of cohesion. Through this especially strong cohesive tendency, water has four unique properties. First, water has incredibly strong cohesion force. Second, water has a very high specific heat. Third, ice is less dense than liquid water. Fourth, water is the universal solvent. The first property, water's strong cohesion tension, causes H2O to move up xylem without using energy. More specifically, transpirational pole cohesion tension occurs as one H2O molecule is lost through transpiration, and another H2O is drawn in at the roots. Cohesion tension also causes water to perform capillary action, which is a combination of cohesion, when molecules stick to other identical molecules, and adhesion, when molecules stick to other dissimilar molecules. Cohesion tension prompts a final phenomenon known as surface tension, which allows insects like the water crawler to literally walk on water without breaking the surface. The second property is water's high specific heat. Cohesion increases the difficulty in separating H2O molecules, and ergo it increases the heat needed for water to vaporize. This also has a great effect on large bodies of water. It allows them to resist temperature change, providing a stable living environment and moderating nearby land climate. The third property, the observation that ice is less dense than liquid water, is due to the hydrogen bonds of H2O molecules. These bonds keep molecules farther away from one another in ice, allowing sheets of ice to form over liquid water in winter. This leads to spring overturn, which occurs when the ice melts, allowing oxygen from the surface to circulate in the body of water and releasing bacterial buildup from the bottom. The fourth property, the observation that water is the universal solvent, is caused by water's high polarity. The polarity allows water to dissolve all polar and ionic substances. Next up are isomers. Isomers are organic compounds with the same molecular formula, but different arrangements or structures. After that, we have the organic compounds. The four main organic compounds, known as macromolecules, are carbohydrates, lipids, proteins, and nucleic acids. With carbohydrates, the monomer is monosaccharide, which are joined by hydrogen bonds to create the polymers, polysaccharides. The two main functions of carbohydrates are energy storage and structural support. Starch and glycogen are the two forms of energy storage for plants and animals, respectively. Cellulose and chitin are the two forms of structural support for plants and animals, respectively. With lipids, the monomer With carbohydrates, the monomer is monosaccharide, which are joined by hydrogen bonds to create the polymers, polysaccharides. The two main functions of carbohydrates are energy storage and structural support. Starch and glycogen are the two forms of energy storage for plants and animals, respectively. Cellulose and chitin are the two forms of structural support for plants and animals, respectively. With lipids, the monomer is a fatty acid and one glycerol, which are joined by covalent bonds to form the polymers, such as triglyceride and phospholipids. The functions include long-term energy storage, protection, and insulation. Examples include saturated and unsaturated fatty acids and steroids. With proteins, the monomers are amino acids, which are joined by peptide bonds to form the polymer, polypeptides. The functions include carrying oxygen, structural support, building tissue, and copying DNA. Examples include muscle tissue and hemoglobin. With nucleic acids, the monomer is a nucleotide, which is joined by phosphodiester bonds to form the polymer, nucleic acid. The functions include storage of genetic info, and examples include DNA and RNA. These organic compounds go through dehydration synthesis to form polymers. First, a water molecule is removed from the adjacent hydroxyl groups. Second, the remaining oxygen joins two monomers with an oxygen bridge. Organic compounds go through hydrolysis to split polymers into monomers. First, water is forced into a bond with the oxygen between two monomers. Second, when the bond is broken, two monomers with hydroxyl groups result from the split. The next topic covers phospholipids, the major component of cellular membranes. The structure of phospholipids includes two parts. First, there are the polar hydrophilic heads of the phospholipid, made with glycerol and a phosphate group. These attract water and other polar substances toward the cellular membrane, both from the internal cell and the external environment. Second are the fatty acid tails, which are nonpolar and hydrophobic. These prevent polar substances and large molecules from easily escaping from or invading the cell. The fatty acid tails are either saturated with a single bond or unsaturated with a double bond. The function of phospholipids and the cellular membrane varies slightly based on its fatty acid tail. Most fatty acid tails are unsaturated, allowing the membrane to remain relatively fluid. Some fatty acid tails are saturated, exhibiting solid pieces of the membrane to provide structural support for the phospholipid bilayer. The final topic in this segment covers protein structure. This begins with the formula of an amino acid. At the center, one carbon is connected to four other components. There's a hydrogen, a carboxyl group, an amino group, and an R or variable group. Then, there are the primary, secondary, tertiary, and quaternary levels of protein structure. The primary level is the unique sequence of amino acids in a chain for one specific protein. The secondary level includes an alpha helix and or a beta helix, which is arranged via the hydrogen bonds. The tertiary level includes the 3D shape, which assigns the protein its specific purpose. The shape is determined by hydrogen bonds and ionic bonds of R groups, hydrophobic interactions, van der Waals forces, and disulfide bonds. The quaternary structure are the proteins that consist of more than one polypeptide chain. This project was in sum generated by Mary Gates.